Hello.
Hello, ma'am.
Hi, ma'am.
How are you all?
Fine, ma'am.
Please switch on your cameras. Risha, you are traveling? Then how will you be able to like attend class?
In bio, probably next 2 classes I'll be missing.
Okay, fine. So I will continue chapter number 15 today. So we have completed before this development topic. So I will be starting from here.
Ma'am, what about the test? Chapter 19 test.
Chapter 19 test I will be taking in last of this week.
Okay, ma'am.
Okay. Okay, and your copies I will be checking in this week only, and I will be sending that answer sheet to you as well. Asmi, have you submitted test copies for both the chapters 17 and 18, beta?
Good evening, ma'am. Ma'am, I have to send the second. I sent the first chapter long back, like the day you gave the test. The next day I completed and I submitted. I didn't know that we had time to, write and all that.
No,
Risha told me.
I have not given time to everyone. I have given time to only Risha because there was some
Oh,
problem with her, no. So I have told her, okay, it's fine if you have some problem. I'm giving you some time, but I have not given time to like every one of you.
I didn't
You
know
all,
that. But I
you all
submitted the
are,
first test long back. The second test I will submit it today. I have completed. I have to just scan and send it to you.
like you all were supposed to submit the test till next day only or till on that day only. From now onwards, the day I am posting the question paper, I want the answer sheet on the same day. No relaxation from now onwards, because I think from now onwards I will taking that I will take that objective test of all the chapters that we have completed till now. So for that I will not give any relaxation. Shreya, you have submitted test copies for both tests, right?
Yes, ma'am. I sent it in the night.
Okay, I have seen that that chapter 18 test copy I have seen, but
Yes, ma'am.
I don't have any idea about 17. I will see and will
Yes,
check.
ma'am. I
Okay,
sent it to you in WhatsApp, both of them.
Beta, why you all are not sending the answer keys on that classroom?
Ma'am, option is not there to attach the file.
Okay, okay, I will see. So Asmi and Anvadya will be coming.
Ma'am, I'll call her and ask.
Please call and ask beta. You and Anvadya are like missing a lot of classes. So I don't know. Shreya, I have to take your classes as well. I think from tomorrow I will be taking your continuous classes to finish your syllabus. That
Okay, ma'am.
first 5 chapters are left, right?
Yes, ma'am.
Okay.
I guess till 8th chapter is left. 8th chapter is done, but then the rest are there.
Seventh is not in your syllabus, right? Seventh or sixth, which chapter is not in your syllabus?
Sixth.
I have started teaching you from chapter number 7, I think. Or 7, I have to teach you till now.
No, ma'am. 7th you have to teach. 8th is done.
That structural organization is 7th chapter, I think, है ना?
Structure and organization is done.
Okay, then chapter number 7 is what I don't know.
I'll check and tell you, ma'am. I'll message all the chapters.
Okay, fine. I'll start with chapter number 2.
Okay, ma'am.
Tomorrow's class, I'll start with chapter number 2, and a small portion of that chapter I have handwritten notes. I will send you those notes. So when I will teach you, please consider those notes because I have not got any time to
Fine,
write
ma'am.
them. Okay.
I'll check them.
Okay. So Asmi, I'm starting class beta. Okay. So what is development? Development is it's a very common term that include all the changes that occur in an organism during throughout their life. Okay. So in plants, different pathways are present. Different pathways occur in response to environment, so that like different organs or different structures can be found. This ability is known as plasticity. So if a question is asked what is plasticity, then you should know that different plant is able to follow different pathways in response to different environmental conditions throughout their different phases of life. So this ability of plant to acquire different structures or different shapes is known as plasticity. For an example, heterophyly in cotton, coriander and larkspur. So what is this heterophyly means? Heterophyly means marked changes in leaf morphology. Marked changes means the changes which we can observe or we can see through our eyes induced by the environmental conditions. Suppose, let's take an example. So here is an example of coriander leaf, hydrophil in cotton coriander. So this is an, suppose this is an leaf of coriander. So initially when the leaf is juvenile, then it looks like that. When the leaf become adult, then start looking like that. Have you, any one of you have observed the leaves of coriander? This is correct? That the initial leaves for coriander are like that, and when the coriander become adult or it's about to like it's ready to get harvested, then its leaves start looking like that, right? Should I proceed?
Yes, ma'am.
Okay, Asmi, don't know Asmi is there or not. Okay, so this is the heterophyly. On the other hand, the leaf may acquire different shapes. It may be because of the environment, and the second thing is that it may be produced in air. Like first example is that the leaf can acquire different shapes when it is juvenile. When it is adult, the shape of the leaves get changed. This is because of the environmental changes. Now, suppose there is a one like different leaves that are that can be produced in air as well as in water. For example, we are taking an example of buttercup plant. So what is buttercup plant? So this is an a plant that can grow in water as well as on land, and it is of medicinal use. Okay, so the leaves of the buttercup plant looks like that when it is present in terrestrial habitat. On the other hand, the plant leaf, the same plant's leaf in water looks like that. So this is because of the shape of the leaves is changing according to the environment in which the plant is growing. Okay. This phenomenon is known as heterophyly. So if one line question is asked what is heterophyly, then you can say the change in the morphology of the leaves because of the change in the environment is known as heterophyly. Okay. Okay. Hi Anavadya. You all are getting my voice? You all are, my voice is audible or not?
Yes, ma'am.
Okay. How are you, beta?
Good, ma'am. How are you?
I am also good. So have you submitted the test copies for both tests, beta?
No, ma'am. I will submit it today after class. Not after.
Oh, beta, I have not given like time to you all. Risha was having some problem, so I told her to take some time. But you all are supposed to submit the test copies on the same day, or अगर कुछ problem है तो you can submit it on the next day. Okay?
Yes, ma'am.
Okay. Please submit it till today night, है ना?
Yes, ma'am.
Okay. So the development, that is growth and differentiation. So can Asmi, can you tell me what is the difference between growth and differentiation, beta?
Ma'am, growth and
Differentiation. In case of plants, there are two terms used for the like development of the plant. First is growth and second is differentiation. So what do you mean by growth and what do you mean by differentiation?
ma'am, probably growth means the little growth of the plant which takes place over the years, and differentiation means the parts is divided into over time. Like, yeah, that's what the time it takes to divide into branches, then the roots, the development of roots and
Differentiation,
flowers.
yeah. Growth means increase in size and differentiation means formation of different organs of a plant.
Yes, ma'am.
Parts of a plant, not organs. Okay. Okay. So both growth and differentiation are under control of some extrinsic and intrinsic factors. So intrinsic factors include intracellular, that is within a cell, genetic, and intercellular, that is between two cells factors. So intra and intercellular factors. Intracellular includes genetic factors, that is within the genes, and intercellular includes some chemicals, just as plant growth regulator. Okay, no. Intracellular includes genetic and intercellular includes plant growth regulator, and light, temperature, water, oxygen, nutrition, they all are also included in intercellular. Okay, extrinsic factors. So there are like 4 or 5 important plant growth regulators we are going to study in this chapter. So some of them are the name of some like PGRs are given here. First is indole compounds, that is indole 3-acetic acid and indole acetic acid. Second are adenine derivatives, that is N6 furfuryl aminopyrin and kinetin. Third is derivatives of carotenoids, that is abscisic acid and ABA. Then terpenes, that are gibberellic acid and GA3, it's also a form of gibberellic acid. And fourth are gases, like some of the PGRs are present in form of gases. It's a very important question. It can be asked that just give a name of the PGR that is formed in the gaseous form, then you should answer either ethylene or C2H4. Okay. Why you all are not switching on your cameras? I'm not able to concentrate. Asmi?
Yes, ma'am.
Beta, please switch on your camera. What happened?
Yes, ma'am. 2 minutes. I'm just trying. I switched it on earlier. I don't know what's happening.
What happened? My voice is lagging.
Yes, ma'am. A bit. I think it's the problem from my side.
Okay. Shreya, you are getting my voice properly, or is there any problem?
It's clear.
Okay, okay, fine. ठीक है. So, like, what are plant growth regulators? They are described. They can be described as this definition you have to learn. They are plant growth substances or plant hormones, or we can say they are phytohormones. So, phytohormones are the hormones that are related to plants only. So, they can be divided into two groups on basis of their functions in the plant. Okay, so first group includes growth promoting first group includes all the PGRs that are involved in promotion of the growth of the plant, like cell division. It causes the increase in size of plant, so cell division. Second is cell enlargement. These are the growth promoting activities that are done by the PGPRs. Okay. And the second group plays an important role in plant responses to wounds and stress such as abiotic and biotic origin. So these PGRS are divided into two categories on basis of their activities occurring. They are doing activities in the plants on basis of that. Okay. So it's clear till now. Should I proceed or is there any problem? Shreya asked me, beta. Anvadya is left.
Ma'am, you can proceed. It's good.
Ashmi is fine, beta?
Yes, ma'am. Yes, ma'am.
Okay, fine. Okay. Okay, so in this chapter, like deeply, we are going to study about the PGPRs that are involved in like both growth as well as in wound healing and everything. Okay, so the important thing is that the third category involves the PGRs that are gaseous. It includes ethylene. So ethylene can fit under both. Means ethylene. It's an important question, beta. It's an exception that ethylene it makes a third category, but it can be grouped under both type of PGRs, that are involved in growth promotion as well as in wound healing. But basically, what is the function of ethylene is that it is it act as an inhibition of the growth activities. It is an inhibitory for growth activities. Okay? Should I proceed? For ethylene, you should remember that ethylene is gaseous, and what's its function? It is inhibitor of growth activities. Okay, it inhibits the growth. So now, how the discovery of plant growth regulators was done, or how they are discovered. So, like it's a very interesting thing to remember that the discovery of these plant growth regulator occurred accidentally. It is not done by any motive. We have to discover the plant growth regulator. They were discovered accidentally. So who discovered them? Charles Darwin, he was then, and his son Francis Darwin, they observed that this is the coleoptile. So what is coleoptile? They are the cylindrical organs that are they are present under the first leaf. So what is the first leaf? What is the other name for first leaf? When we put seed into soil, then what is the first thing that comes out of
Primarodia something.
That is they are the like parts from where the first leaf will start. So tell me, I'm expecting from at least from Asmi and Anvadya, beta, please answer. What is the first leaf? Because I have not taught that chapter to Shreya till now, so I don't think she will be able to answer, but you both should answer. What is the first leaf that comes out of the soil when we put seed into soil? Please answer it.
Ma'am, from the seed to the plant.
Yeah.
Am I talking about the cotyledons?
Yeah, Navadya, please.
Cotyledons.
Yeah, very good. Cotyledon. Cotyledon is known as the first leaf, and on basis of that, there is a difference between monocots and dicots. I told you earlier. You remember that?
Yes, ma'am.
Okay, fine. So this is this region that is present just below the first leaf and the shoot apex in grass is known as coleoptile. That cotyledon is not applicable in this case, but I asked you general question. कि हम first leaf किसको कहते हैं? That cotyledon is known as, but here it is known as coleoptile. Okay, in case of grasses. Now, so what they observed that these coleoptile that are present at the tip of canary grass, they responded unilateral illumination by growing towards a light source that is known as phototropism. So what is phototropism? When he observed, when he put this grass in front of light, or then he observed that the tips of the grass start moving or bending towards the light. Okay, so this was an experiment done by him. It's not very important. You just have to remember that the tip of the collector will start moving towards the light. तो इससे discovery हुई आपके different PGRs की. So okay, these all about this all thing is about scientist name and all. So I don't think I should read this thing. Or if you want, then I can read it for you all. These are discoveries of different scientists who discovered different these PGRs. So you have to learn this because it's very important. Name of the question can also be name of the scientist can also be asked, like who discovered indole acetic acid and who discovered ABA. So you have to answer. So there is nothing like to explain. This is just to learn. So should I read it or should I proceed?
Ma'am, you can proceed.
Fine, Asmi and Shreya?
Yes, ma'am.
Okay. Now, what are the physiological effects of plant growth regulators? It is very important and interesting topic, and I think for this year, this is the topic that is present in your syllabus too, right?
Which one, ma'am?
This plant growth regulators. From this chapter, only this thing is present in your
Yes, ma'am.
syllabus. Right?
Only the hormones, yeah.
Hormones, and they are very interesting too. Okay, so first hormone is auxin. So what are the things you have to remember about auxin is that the auxin was isolated for the first time from the human urine, and this auxin, the other name for auxin is indole 3-acetic acid, and it is also known as IAA. A question can be asked, and I think question will be asked, that what is the other name for auxin, and you have to answer its other name is indole 3-acetic acid, and in bracket you have to write indole acetic acid. Okay, so auxin is also known as indole acetic acid, and okay, so they are generally produced. It was first isolated from human urine, but they can be also produced by the growing apexes of stem and roots. So you know what is the apex of root and stem? From the tip of root and stem they can be produced, and but they will not work there. These auxins will be produced at the apex of the stem and roots. From there they will migrate to the regions where their function is required, or they function at some other area, but they are produced at the apexes of the stem and roots. Okay, and if a question is asked, name one or two, like two auxins that are isolated from plants. So you have to remember that indole acetic acid or indole 3-acetic acid and butyric acid are the two auxins that are produced from the or isolated from the plants. Okay, and the third type of auxin is naphthalene acetic acid and 2,4-D, that is 2,4-dichlorophenoxyacetic phenoxyacetic. They are synthetic auxins. So if it is asked, name two auxins that are produced naturally and two auxins that are produced artificially or synthetically, then you have to answer that natural auxins are indole acetic acid and butyric acid because they are produced from plants, and the synthetic auxins are NAA, that is naphthalene acetic acid, and 2,4-D, 2,4-dichlorophenoxyacetic acid. They are synthetic auxins. Okay, and what are the use of the auxins? They are used in agriculture and horticulture practices. And what is the main function of these auxins? Why they are used in agriculture and horticulture? Because these auxins are basically important for initiating rooting in stem cuttings. Okay, you know that many plants like rose, they grows by stem cuttings. So how a stem is able to regrow into a new plant? So the auxin that will be produced in the tips of that root cutting will produce auxin that will be able to initiate the rooting in that stem. Okay. Oh, where is Asmi now? Okay, so and one more thing is that these auxins are able to produce promote flowering in pineapples as well, and they help to prevent fruit and leaf drop at the early stages, but they promote the abscission of older mature leaves and fruits. So it's a very important and interesting thing to remember about auxin is that it prevents the drop of leaves and fruits in the early stages, but as the fruits and leaves become yellow or older, then they are responsible for abscission of the older parts of the leaves, older parts of the tree like leaves and fruits. Okay. And one more important thing is apical dominance. So this it is the most important thing to remember about like auxin is that it is involved in an important phenomenon known as apical dominance. So what is apical dominance? Apical dominance means the growing apical bud inhibits the growth of the lateral buds. It is maintained or regulated by auxins only. Okay, and I think I have added a definition for that apical dominance as well. So this is the apical bud and these are the little buds. So auxin is, you must have observed that the apical region or the apex of the plant keeps on growing, but the little buds have a limited growth. This is because of the auxin only. Auxin is responsible for controlling the growth of the apical bud, but a limited growth of the little buds. Shreya and Anvati, you are getting my voice, right?
Yes, ma'am.
Yes, ma'am.
Because your cameras are not on, so I'm not able to understand that like the video is audible or not. Okay. So auxin are also, so the main two things we have to remember about auxin is that about what are natural auxins, what are synthetic auxins, their functions, and what is apical dominance, and they are also responsible for inducing parthenocarpy. So Anvadya, can you tell me beta, what is parthenocarpy? What is parthenocarpic fruit? Shreya, if you know, you can tell me beta. It's not true fruit जो fertilization से नहीं बनेगा. You remember that partheno? Yeah. Your voice is on mute, beta. You're on mute. I'm not able to hear your voice, beta. Still your voice is not audible, beta. You are on mute.
Oh, ma'am.
Yeah.
The screen went black. It's fine now. Ma'am, it's like fruits that are naturally made or something like that.
No, that are made naturally without like fertilization के जो fruits बनते हैं, they are parthenocarpic fruit. Just go like after class, search on Google what are parthenocarpic fruit, because seeds, fruits without seeds are known as, I think, parthenocarpic seedless fruits. Or just if you have mobile, just search what is parthenocarpic fruit. I forgot exact definition about that. Seedless fruit, I think they are.
Yes, ma'am, they are seedless fruit.
Yeah. Okay. So auxin is clear. Should I proceed?
Yes, ma'am.
Yes, ma'am.
So the second are gibberellins. So they are again another type of PGRs, and they are present in many forms like GA1, GA2, GA3 and so on. So basically this gibberellic acid, that is GA3, it was one of the first gibberellin that was discovered and is most studied form of gibberellins also. And the important thing to remember about gibberellins are they all are acidic, okay? And they produce a variety of physiological response in plants, like they are able to cause an increase in the length of the axis, and for an example, they're responsible for increasing the length of grape stalks. So auxin ka main function is to initiate the rooting, okay? And what is the important function of gibberellin is that they are responsible for increasing the length of the axis or stalk or stem ka length ke liye they are responsible. And these gibberellins causes fruits like apple to elongate and improve its shape. They also delay the senescence, that is the aging, okay? So these are the functions gibberellin is performing. All of these functions you have to remember. So do one thing, make a table. What are gibberellins, what is their common name, make 3-4 columns, name of the hormone, and what is their common name, what are their function, and who discovered them, and from where they are discovered. If 5-column table you have to make and copy paste the things into them and learn that table. Otherwise ये ऐसे पढ़ने बैठोगे तो you will get confused between the functions of each PGR, okay? Fine. So
Yes.
because they are able to delay the senescence, means they prevent the aging of the plant, so the fruits can be left on the tree for longer time so that they can be used as it is required. So suppose if an injection is like gibberellins are injected into a plant, then what will happen? Suppose there is a tree of apple. So it get flowering, like flowering start in the complete full tree. The flowering starts at the same time. So the fruits will also we get at the same time, but we don't want to get fruits on the same time because we want to sell them from time to time. So a particular portion of the plant is given the treatment of gibberellin so that the apples that we are getting from that particular part or from a particular tree we will get after some time as we require. So it is used for delaying the senescence in the plants, okay? Now sugarcane, another example is in case of sugarcane, sugarcane stores carbohydrates in form of sugar in their stem. So if a sugarcane crop is sprayed with gibberellic acid, then it increases the length of the stem, as a result of which the yield of the sugarcane will also be increased, okay? So this gibberellin is also complete. One more thing is that these gibberellins are also important, are also playing an important role in promoting bolting. So what is bolting? That is increasing the internodal elongation just prior to flowering, okay? Internodal means that is, suppose this is a sugar cane, this is one node and this is another node. So the region that is present between two nodes is known as internode. So the internode portion is increased, because of that the length of the sugarcane plant is increasing. So as the length increases, we will get more amount of sugarcane, okay? So it's clear. Should I proceed or is there any doubt, Shreya and Vadya?
No doubt, ma'am.
Now we can proceed.
Fine. And Anavadya, please text me which chapter your ma'am started after this chapter so that I can make notes for that chapter. I think this chapter will be completed today only. Do you have any idea which chapter your ma'am started after this chapter?
Ma'am, give me a minute. I will tell you what chapter she's.
It's fine. After class, you can text me, but please text me because
Yes,
I have
ma'am.
to make notes of that today only.
Yeah.
Okay.
Yeah. Yes.
Shreya, till which chapter like syllabus is complete in your school, beta?
Ma'am, biomolecules.
Oh, so biomolecules is running in your school, right?
Ma'am, biomolecules is done. The next chapter ma'am is doing. She took a test of biomolecules today.
So you understood anything about biomolecules?
Yes, ma'am, it was clear.
Okay, so if I have any doubt in biomolecules, I will ask you.
Okay, ma'am.
Because like that chapter is quite tough for me, because that is like thoda sa it's related to chemistry, right?
Yes, ma'am. She gave 30 objective type questions for that chapter from the textbook.
Okay, okay, fine. ठीक है. Okay. So cytokinins, the third type of PGR is cytokinins. They have specific effects and they were also discovered as kinetin. So what is the another name for cytokinesis is kinetin. Okay, that is a modified form of adenine and purine, and like they are again obtained from herring sperm DNA. So these things about cytokinin and auxin you have to remember, that the auxins are also known as IAA or indole 3 acetic acid, and they were obtained from human urine, and these cytokinins are also known as kinin, and they were they were obtained from herring sperm DNA. Okay, these things you have to remember because question can be asked on that. Okay, so what are the functions of cytokinin are? Okay, these cytokinins naturally can be synthesized in regions where rapid cell division occurs. For example, root apexes में meristematic activity will be present, so cell keeps on dividing because meristematic cells will be present there. So these root apexes are able to produce cytokinins. Don't get confused between the apexes. वो auxin was present at the shoot tip and root tip. Okay. Auxin was synthesizing at the shoot tip and root tip or apex of the shoot and root, and these cytokinins are produced at the root apexes and developing shoot buds. Okay, small buds, small shoots that are developing, young fruits, etc., and they help in production of new leaves, chloroplast in leaves, lateral roots, lateral shoot growth, and adventitious shoot formation. So these cytokinins helps in overcoming the apical dominance. One more this is responsible for this is involved in overcoming apical dominance, and which plant hormone like plant growth regulator is involved in creating apical dominance?
Auxin.
Yeah. So auxin is responsible for creating apical dominance, means it helps the apical bud to grow and later buds to stop or reduce their growth. But that apical dominance can be removed with the help of cytokinin treatment. So if a question is asked, what is apical dominance? Which PGR is involved in creating dominance, apical dominance, and which is the PGR that is involved in overcoming or removing apical dominance, then you should know the question for all these three questions. You should know the answer for all these three questions. So right now you are able to answer these three questions, Shreya and Anvadya. What is apical dominance? Apical dominance is a phenomenon where the apical portion or apical bud keeps on growing, but the lateral bud's growth is stopped or slowed down. And which PGR is responsible for creating apical dominance? Auxin. Which PGR is responsible for overcoming apical dominance? Cytokinin. Okay?
Yes, ma'am.
Yes, ma'am.
Okay. So what is apical dominance? Answer I have added here also. It's the phenomenon where the main or the central stem of the plant is dominant, means it keeps on growing over the lateral branches of the stem. So this is the definition for apical dominance. So it's clear till cytokinin. Should I proceed with ethylene?
Yes, ma'am.
Fine, Shreya beta.
Yes, ma'am.
Okay, so what is ethylene? It's, as I told you earlier, it's a simple gaseous plant growth regulator, and it is synthesized in large amount by the tissues that undergo senescence, means aging. Aging के लिए इसलिए this ethylene is used to it's used for ripening of fruits. Okay, fast ripening of fruits is done with the help of this ethylene. Okay. So these are produced in the area, in the regions or in the tissues that undergo senescence and ripening of the fruits. And influences of ethylene on plants includes horizontal growth of seedling. Okay, so the seedling means the small portion of the seed that get germinated, and the first portion, small plant, is known as seedling. So till now we have studied all the hormones that are responsible for increasing the length, like cytokinase is responsible for increasing the length of the plant, but this ethylene is responsible for increasing the horizontal growth of seedling. Okay, means यहाँ एक one seedling 2, 3, 4 they are responsible for increasing the horizontal growth, buds निकल के, and swelling of the axis and apical hook formation in dicot seedlings. And this apical hook, I think I have not added anything related to that, but I will teach you in next class that what is this apical hook formation. It's a very important phenomenon. I don't know how I skipped this thing, but I will add and will let you know in next class. Please remember that I have, wait 1 minute. It's a very important phenomenon, and question can be asked on this. What is this apical hook formation in dicot seedling? It's done by ethylene. So this ethylene is responsible for promoting senescence and abscission of the plant organs like leaves and flowers. So it is effective in fruit ripening if it is injected or sprayed on the fruits that are unripened, and then they will be ripened very fast by the effect of this ethylene. Because how this ethylene enhances the respiration rate during the ripening of the fruit. So if it is asked which PGR is given to unripened fruit so that it get ripened fast, then you have to reply ethylene. And how it works, this ethylene enhances the respiration rate, and as a result of which the fruit get ripened fast. And this process, this phenomenon of increase in rate of respiration is known as respiratory climactic. Okay. And the ethylene is also responsible for breaking down seed and bud dormancy. Seed dormancy and bud dormancy, you know. What is seed dormancy? Seed dormancy is a phenomenon where the seed will remain under the soil, will not germinate in unfavorable conditions. Suppose if there is a plant or the seed that is that is supposed to grow in winter, and the seeds are still there under the ground, but the situation is not favorable. Like in summer, the winter plants can't grow. Okay, so during that time, the seed will remain in dormant period. Means all the metabolic activities will be ceased in that, but it will like survive, but it will not grow. That is the phenomenon known as seed dormancy. Similarly, bud dormancy is also there. So when the favorable condition come, then that seed will convert into seedling and will eventually grow into a complete plant. Okay, so this ethylene is capable to break the seed dormancy and bud dormancy. Okay, and this ethylene is also like responsible or helpful in promoting the growth of the root and the root hair formation, as it is responsible for increasing the it is responsible for growing root hair. So they are helpful in increasing the absorption area. We know that these root hairs are responsible for increasing the absorption area or absorption surface for the root. I told you in some earlier like chapters, I told you what is the role of these root hairs. Remember, Anvadya? Root hairs are responsible for increasing the absorption surface for the roots to absorb the minerals and water. Okay. So if like the ethylene is one of the like PGR that is involved in variety of physiological processes. So this is the most widely used PGR in case of agriculture, or agriculture fields are using mostly this ethylene. So the most common compound that is used as the source of ethylene is ethifone. Again, a question can be asked on this, which is the most commonly used form of ethylene in agriculture field, then you should know it is ethifone. So what is this ichthyophon? It is an aqueous solution and is readily absorbed and transported within the plant and release ethylene slowly. So what is ichthyophon? It is a type of ethylene or source of ethylene that is used commonly as a ethylene source in agriculture field, and it is an aqueous solution. Aqueous solution means it is a solution with water. It can be absorbed easily and transported within the plant, and it releases ethylene slowly as required. Okay, so ethylene is also clear. Okay, now the next is abscisic acid. The abscisic acid ABA, it was discovered for its role in regulating abscission and dormancy. Okay, so abscission, what is abscission and dormancy? Abscission is again like falling off leaves and fruits is abscission. कोई भी चीज when it get ripened, then it start abscission and senescence. Okay. But like other PGRs, it also has other wide-ranging effects as in form of plant growth regulators and development. So what is the main function of ABA is that it acts as general plant growth inhibitor and an inhibitor of plant metabolism. 1 minute. Yeah, so okay, so this abscisic acid, it inhibits seed germination. This is the most important function performed by ABA. It stimulates the closure of stomata and increases the tolerance of plants to various kinds of stress. So what are stomata? They are the pole-like structures that are found on the epidermis of leaves and stems, and basically they are involved in exchange of gases. When like the plants are taking CO2 and for cooking their food and are releasing oxygen in turn. You all know that, I think you know that. So this process of taking CO2 and releasing oxygen is occurring with the help of the stomata, and these ABA are responsible for stimulating the closure, not opening, closure of stomata, and they increases the tolerance of plants to various kind of stress as well. Okay? Therefore, it is also known as stress hormone. So if question is asked out of the following, which is known as a stress hormone, then you should know ABA, abscisic acid. And it also plays an important role in development of seed, maturation of seeds, and seed dormancy. So, but as they are involved in inducing dormancy, these ABA helps the seeds to withstand desiccation, bursting. Desiccation means bursting, and other unfavorable conditions for growth. So mostly these ABA acts as an antagonist to gibberellic acid. So if a question is asked like which out of the following, which hormone is antagonist to ABA, then you have to answer gibberellic acid. Why? Because they are responsible for inducing dormancy, and that gibberellic acid is responsible for like preventing dormancy. They are creating dormancy, and they are inducing dormancy, and that are breaking dormancy. Gibberellic acid. Okay.
Yes, ma'am.
Yeah, ABA is responsible for inducing dormancy, means they helps the seed to survive in unfavorable condition. They will induce dormancy so that the seedling can survive but not growth. But gibberellic acid are responsible for breaking down dormancy. Okay? What is dormancy? It is the period of arrest of plant growth that enables the plant to survive in unfavorable condition, but it will not grow. Okay? Okay, so this ABA is also involved in some 1 minute, huh? Genomic control of antigens, important one. So along with these, many extrinsic factors like temperature, light, They control of plant growth and development also occur with the help of plant growth regulators. So like there are some features or some events like vernalization, flowering, dormancy, seed germination and plant movement. They all are regulated with the help of these different PGRs. So what is vernalization? Vernalization is a process that involves induction of flowering process in plant by exposure to the prolonged cold of winter or by an artificial equivalent. So what happened? The plant is kept in very cold temperature so that the flowering can be induced. So either the temperature should be very much cold or the cold temperature can be produced or created artificially to induce the flowering. It's a kind of like method or process for induction of the flowering using different PGRs. So we will like I think in detail we will learn this vernalization in this chapter. So how much time is left?
Ma'am, I think 40 minutes.
Okay, but I think I will be going to finish this chapter within 10 minutes. Let's see. Okay. So you are understanding Anvadya and Shreya, right? I'm not running very fast.
Ma'am, it's okay. We can understand.
Yes, ma'am.
Fine, Anvadya?
Yes, ma'am.
Phototropism. So what is phototropism? Like it's a phenomenon the plants require a certain period of light or certain amount of light is required by each plant for specific period so that they can induce flowering. So we can see that there are some plants that are able to like it's an internal thing that is fixed in the plant. It like the plants are able to measure the duration of exposure of light. There are some plants that require 1 to 2 hours light only, but on the same hand at the same on the other hand there are some plants that require a long period of light, means they require full day light. Some are the plants that require very less light. So on basis of that the plants can be categorized into two type of like categories: short day plant and long day plants. Long day plants are the plant that require long period of sunlight, whereas short day plants are the plants that require very less amount of light and they blooms in the night. So some plants require exposure to the light for a period exceeding a well-defined critical duration. So what is this critical duration? Critical duration means that the time that is required, that the time like the light that is required for a specific time by the plant. So this critical duration will be different for different plants. Okay, critical duration of light means particular time for what the plant is requiring light. Okay, but there are some plants that must be exposed to light for a period less than its critical duration before flowering initiated in them. So on basis of this there are long day plants that require more amount of light, whereas short day plants that require less amount of light for flowering. So there are many plants, like three categories of plant will be there: long day plants that will require more amount of sunlight for flowering, short day plant that will require small amount of light for flowering, and there is third category where there is no such correlation between the exposure of light duration and induction of flowering response. These plants are known as day neutral plants. So on basis of like requirement of light for the process of flowering, the plants can be divided into three categories: long day, short day, and day neutral. Long day will require more amount of light, short day will require less amount of light, and they will bloom in night. And the day neutral plants will not depend on the light. They will grow independent of light on its own. Okay, fine. Okay. Okay, so not only the light period, but also the duration of dark period is also important for inducing flowering in the plants. So the response of plants towards the periods of day and night is known as phototropism. So what is phototropism? It is a like phenomenon where the plants are responding to different periods of day and night is known as phototropism, and it is playing a very important role in flowering process. Okay. So, but it is very interesting to note that the shoot, although the shoot apexes, they modified themselves into the flowering apexes prior to flowering. You know that the shoots are responsible for developing the flower buds that will develop into flower. But
Ma'am, ma'am, can I join Akash?
yeah, okay, beta, bye.
Thank you, ma'am. Bye.
Okay, bye. But it's very interesting thing to note that the site of perception of light and dark duration are the leaves. So like the phenomenon of flowering is directly indirectly controlled by leaves, although the shoots are responsible for developing the buds and the flowering will occur. But the phenomenon or the site of perception of light and dark, sunlight and darkness is received by the leaves. So it is hypothesized that there is a hormonal substance, there are some hormones that is responsible for controlling flowering. This hormonal substance migrates from leaves to the shoot apexes for inducing flowering only when the plants are exposed to necessary inductive photoperiod. So we know that the flowering will occur on the shoots, but the induction of flowering is controlled by the hormones that are produced in the leaves in response to the light or dark. Okay? And whether it's confusing or it's clear, beta?
It's clear, ma'am. Basically, that's a hormone in the leaves that induces flowering.
Yeah, in response to light and dark. Like so if it's long day plant, then it will require very much sunlight, and in response to that sunlight, some hormone is produced within the leaves that will migrate from leaves to shoot and will induce flowering. Okay? Okay. So the next topic is vernalization. So what is vernalization? There are some plants for which flowering is either quantitative or qualitatively depend on exposure of low temperature. Quantitatively means how much amount, and qualitative means what is the quality. Means like low quality, higher quality. So qualitative is related to that quality and quantitative is related to quantity. So the flowering in case of some plants depends upon the quality and quantity of the time that is the particular plant is kept under low temperature. So suppose there is one plant that is kept in low temperature for 2 days, then flowering started in them. On the other hand, there are some plants which we kept under low temperature for 1 week, then flowering is induced in that. So the process of developing or inducing flowering in plant by keeping them in low temperature is known as vernalization. Okay. So this vernalization refers specifically to the promotion of flowering by a period of low temperature. Some important fruit plants like wheat, barley, rye, they have two kinds of varieties. One is winter variety and the second is spring variety. So these spring varieties are normally planted in the spring season and come to flower and produce again before the end of growing season. But the winter varieties, if they are plotted in the spring, then would normally fail to flower and produce mature grain within the span of flowering season. So they are planted in autumn. Autumn में उनको plant करके उनको उनके seeds को they are kept in low temperature so that the flowering can be induced in them when the conditions are favorable. Okay? कुछ varieties होती है जिनको like in year they are shown for the 2 time. ठीक है? So एक तो favorable period होगा when it can be grown naturally in the fields. One is like the planting time is favorable, but they are not harvested at the same time. They are kept under cold temperature or cold storage when they are kept, so that they can be released when the season is over, off season mein, to provide varieties. So these plants that are planted in autumn, they get germinate and over winter come out as small seedlings. So they will be planted in autumn, they get germinated, and all the time during winter they come out as small seedling, and they resume growth in the spring, and they are again harvested usually around the midsummer. So the seedling, unke seeds ko they are keeping them under cold storage. So autumn में they are again planting those seeds. तो winter के time में they start growing, and जब तक उनका harvesting का time आएगा, summer have arrived. So it's clear, Anuvadya?
Yes, ma'am.
Okay. So most important example of fertilization is seen in case of biennial plants. So there are three types of plants: annual plants, biennial plants, and perennial plants. Annual plants are the plants whose products can be seen once in a year. We can, for example, methi, fenugreek. We are getting good quality of methi in winter season only. Okay, so it's an annual plant because we are getting once in a year. Biennial plants are the plants whose we can say crop we are getting twice in a year. 6 months we will get, then another 6 months we are also able to get. And there are some perennial plants whose crop we are able to get throughout the year or within 3 months. After every 3 months we are getting the fresh stock of the same crop. They are perennial. So in biennial plants this vernalization process is seen. Okay, then the last topic, I think it's seed dormancy. So when there are certain seeds that fail to germinate even when the external conditions are favorable, these seeds are understood to undergo a period of dormancy that is not controlled by any external environment, but it is controlled by the seed itself. There are some endogenous conditions within the seed itself that is maintaining that dormancy. Suppose there are some seeds lost their viability if they are kept for very much time, long time, then they fail to germinate, or their capacity to get germinate is over. Then this phenomenon is known as permanent dormancy, because that seed is not able to grow into a seedling. Okay, that impermeable and hard seed coat that is present, that can be that can be broken down with the help of certain chemical inhibitors like abscisic acid, phenolic acid, para-ascorbic acid, and immature embryos. They are some reasons that causes the seed dormancy. Okay? So this seed dormancy can be overcome by natural means also, if we do what we can say scarification of the seeds and sow them in the put them under the soil, then they will be able to grow. And there are many man-made measures as well, like seed coat barrier in some seeds can be broken by mechanical abrasions using knives, sandpaper, etc., or vigorous staking. In nature, these abrasions are caused by microbial actions because the seeds are kept under the soil. So there are many microbes present within the soil that can do the breakdown of the dormancy in the seed. Okay, so and the inhibitory substance that prevents the seed to prevent the seed dormancy can also be done by giving seeds the chilling conditions or by application of certain chemicals like gibberellic acid and nitrates. So environmental conditions, if they are changed like temperature and light, they can also help in removing seed dormancy. So now this chapter is complete. So is there any doubt, Anuvadya beta?
No, ma'am.
Like functions of each PGRs you have to learn. Just make a table in the way I told you. then you will be able to learn about each PGRs. Otherwise, there will be like you will get confused. Okay, and please text me about the next chapter.
Ma'am, currently currently our ma'am is doing, 1 minute, the chapter 13, photosynthesis in higher
Okay,
plants.
okay, so before that, one more chapter is done, right?
Yes, ma'am, but that I have to find out. Today, like she was
Okay, so
doing
I'll start. Okay, fine. So I will start the chapter 13 in next class.
Okay, ma'am. Yeah.
Okay?
Yes, ma'am.
Okay. Okay. Okay, bye.
Bye, ma'am. Thank you.
Bye.